So today we're going to be talking about cellular respiration, which is basically how cells of organisms convert, energy from nutrients into ATP. And cellular respiration can be broken into three steps. First, we have glycolysis, and then we have Krebs cycle, or also called citric acid cycle, and then lastly, ETC, or electron transfer chain. First up, we have glycolysis, which happens in all living organisms, takes place in the cytoplasm of the cell, and requires no oxygen. The 6 carbons get split into 2 using 2 ATP, which then turns into 2 ADP. The P's bond to the carbons and create pGal. pGal then turns into pyruvate, which creates energy. NAD comes into play to pick up the hydrogen, turning NAD plus into NADH on both sides. The products of glycolysis are 2 net ATP, 2 NADHs, and 2 pyruvate. Next up, we have Krebs cycle that also goes by citric acid cycle. This takes place in the mitochondria of eukaryotes and requires oxygen. Before the Krebs cycle, we have the transition step, which is required before entering the cycle. Pyruvate are going to enter the matrix of the mitochondria, but pyruvate itself cannot enter the Krebs cycle, so the transition is turning pyruvate into acetyl-CoA. Acetyl-CoA is a 2-carbon sugar, so a carbon is going to break off from each of the pyruvates. Those 2 carbons added with oxygen make CO2. 2 pyruvates are now 2 carbon sugars, which get coenzyme A, which then makes 2 acetyl-CoAs. We have the Krebs cycle now. Acetyl-CoA can now go into the Krebs cycle. The carbons with the oxygen end up as 2 carbon dioxide. In the Krebs cycle, every time we go around, 1 ADP goes to 1 ATP, and also 3 NAD pluses turn into 3 NADHs. FAD comes into play with 1 FAD plus turning into an FADH, and all of this happens twice in the cycle. The products of the Krebs cycle are 2 ATP, 8 NADHs, 2 FADHs, and 6 carbon dioxide. The last step of cellular respiration is the ETC, also known as the electron transfer chain, which happens through the cristae of the mitochondria. In this step, we have a phospholipid bilayer. The end goal of this process is to get ATP synthesis moving and working. This generates ATP once it starts working. ATP synthase needs a positive proton gradient. And if there are more hydrogen protons in the inner membrane space, the ATP synthase will start moving them back into the matrix, generating ATP in this process. So the NAD and the FAD have the hydrogens to deliver. Hydrogen atoms have 1 electron and 1 proton. These electrons do a little dance while they move through the pumps, which gives off enough energy to pump the hydrogen protons into the intramembrane space. 2 FAD plus turns into 2 FADHs through this as well. Protons cannot be left in intermembrane space though, because they're acidic, and this won't end up very well. So the ATP synthase will move protons back down, thus creating ATP. The final products of the ETC are approximately 30 ATP and 6 waters.